Welcome, my dear students. So far, we have studied about characteristics of a living organism and how classification system was evolved. In today's lecture, we shall study features of organisms and classification systems. In this lecture, we shall study classification systems in detail. First of all, whether we are looking at a plant, an animal, or microscopic bacterium, every single living cell on Earth shares three fundamental components. Think of these as the basic survival kit for life at the microscopic level. First of all, we have the cytoplasm. This is a jelly-like substance that fills the interior of the cell. It is not just filler, though it acts as the vital medium where most of the cell's metabolic chemical reactions actually take place. So next one is the cell membrane. This is a semi-permeable lipid bilayer that encloses the cell. Its job is to maintain homeostasis by acting as a security guard, carefully regulating exactly which substances like nutrients or waste are allowed to enter and exit. And finally, we have DNA. As we discussed in classification, DNA deoxyribose nucleic acid is a universal genetic code. It provides the instruction manual that every cell needs for growth, development, and reproduction. If DNA is the instruction manual for life, then ribosomes are the builders. They are arguably the most important organelles to discuss because without them, the instructions in our DNA would never actually become anything real. So ribosomes are tiny but mighty. You can find them in two places within the cell, freely floating in the cytoplasm, attached to a structure called rough endoplasmic reticulum. We shall also write that as RER, means rough endoplasmic reticulum. Their primary function means the primary function of ribosomes is protein synthesis. Ribosomes read the genetic instructions sent from the DNA and use them to build proteins. But why do proteins matter so much? First of all, they build your tissues like muscles and the internal skeleton of your cells. Secondly, they act as enzymes, which are biological catalysts that speed up chemical reactions. And lastly, they transport molecules and send signals between cells. Basically, ribosomes are the reasons we have a physical body that functions. Here, this is an important note for IGCSE exam and even for A levels, that it is very helpful to remember that while all organisms have ribosomes, they differ in size. We shall study these size differences in very detail in our next lectures, but here I want to only focus on two different size, that is 70S and 80S. In prokaryotes, those have smaller 70S ribosomes while eukaryotes have larger 80S ribosomes. This is a very common compare and contrast exam point. For a long time, the word of biology was very simple. If it was green and stayed still, it was a planet, it was a plant. If it moved and ate things, it was an animal. But as our tools improved, we realized that nature does not like to stay inside such small boxes. So there, the kingdom evolution started. Historically, we used a two kingdom system. The first one is kingdom Plantae, and second one is kingdom Animalia. Kingdom Plantae, that is also named as Regnum Plantae, and kingdom Animalia, Regnum Animali. Okay, that system was proposed by Carlos Linnaeus in 1758. Okay, this binary system uh, for, this binary system broke down as soon as we started looking closer. For example, in kingdom Plantae, this was for organisms which are green and stationary. But kingdom Animalia was used for organisms that move and consume others. This binary system broke down as soon as we started looking very closer. We found organisms that defied these simple definitions. For example, fungi. They stay still like plants, but they do not photosynthesize. They eat by absorbing nutrients, just like animals. But bacteria, those are tiny, single-celled organisms with no nucleus. They clearly are not plants or animals. And the last one, which is the rule breaker, that is Euglena. Some microscopic organisms like Euglena can swim around like an animal, but also photosynthesize like a plant. So this two kingdom model failed because it ignored cellular structure and modes of nutrition. To be biologically meaningful, we need more drawers to sort out the incredible diversity of life. Here is an important tip for you students that IGCSE and A-level examiners often ask why the two kingdom system was replaced. The most accurate and the model answer usually involves mentioning the discovery of prokaryotes, that is bacteria, and the unique chitin cell walls of fungi. We shall study that in detail in our next slides. So to make our drawers of life more accurate, scientist Robert Whittaker proposed a new system in 1969. Instead of just looking at if something moved, he looked at two things: cell structure, that if either there is a nucleus or there is no nucleus, and secondly, mode of nutrition, that how does it get its food. This gave us the five kingdoms of life, each with its own unique set of rules. The first one is prokaryotes, that is Monera. These are the simplest organisms which are single-celled, and most importantly, they lack nucleus. Remember this thing. Prokaryote means no nucleus. Bacteria are the classic example here. The second one is kingdom Protista, and we call those organisms as protoctists. This is often called the odds and ends kingdom. They are mainly unicellular eukaryotes. They do have a nucleus. For example, amoeba and chlamydomonas. Third one is fungi, mostly multicellular heterotrophs, meaning they eat other things. Their unique feature is a cell wall made of chitin. Okay, remember, the cell wall of fungi is made of chitin. This includes everything from giant mushrooms to tiny yeast. And the last one are plants and animals. Plants, as we have discussed earlier, these are multicellular autotrophs that make their own food via photosynthesis. They have cell walls made of cellulose. Remember this difference. Fungi, they also have cell walls, but that is made of chitin, and plants, they have cell walls, okay, that is made of cellulose. And the animals, which are also multicellular but heterotrophs, they lack cell walls entirely. They have no cell wall, allowing for much more complex movement and specialized tissue. By breaking life down into these five groups, we can see exactly where an organism fits based on its internal blueprint. We like to think of the five kingdoms as neat, separate boxes, but nature does not always follow our rules. This creates what we call taxonomic tension, where an organism seems to belong in two places at once. For example, protophyta puzzle. Some protoctists contain chlorophyll and photosynthesize, making them look exactly like plants. This leads to the algae debate. Many algae are unicellular or very simple multicellular organisms, which technically makes them protoctists. However, because they have the same green pigments as plants and make their own food, some scientists argue they should be in the plant kingdom. The lesson here is that classification is a human-made model trying to describe a continuous spectrum of evolution. Because life is always changing, these boundaries are often the subject of intense scientific debate. So if you are asked about to distinguish between a protoctist and a plant, the keyword is differentiation. Plants have specialized tissues like roots, stem, and leaves, while algae and protoctists, even the multicellular ones, lack these complex tissue systems. Remember, no specialized tissues, then that is probably a protoctist. So complexity is the deciding factor. Now that we have seen how hard it can be to draw the line, let's look at a group that is so unique. But before that, look at this picture for comparison. This is alga and plant. Okay, unicellular alga. This is a picture of a clematomonas, and on right side that is a multicellular plant. You can see very clearly the difference of complex tissues. In unicellular alga, there is no complex tissue, that is protoctist, while in multicellular plants that have multiple tissue complexes. So let's look now next to a unique group. It is almost impossible to classify. That is the viruses. If you look at five kingdoms, you will notice something is missing there. That is viruses. Even though they can make us sick and they have their own DNA, biologists describe them as being at the edge of life. In fact, they are not even included in any kingdom. Why are not they considered alive? That is a question arises in our mind because they lack the universal cellular machinery that every other life form possesses. For example, no cell membrane. They don't have their own lipid bilayer. If they have a coat, they usually steal it from a host cell. The number two is cytoplasm. There is no internal fluid for chemical reactions. And the third one, there is no ribosome in virus. They cannot build their own proteins. They have to hijack a host factory to do it from. So when we look at Mrs. Gren, MRSGREN, if you remember the first lecture, that is characteristics of life, movement, reproduction, sensitivity, growth, respiration, uh, excretion, and nutrition, which are the seven processes of life, viruses fail almost every test. They do not respire or produce their own energy. They do not grow in size or excrete waste. They only reproduce by invading a living cell and forcing it to make copies of the virus. So virus is essentially just a bad news message wrapped in a protein coat. They are biological hitchhikers, completely inactive until they find a host to do the work for them. This is where it gets very confusing. If a virus has DNA or RNA, which is the code of life, how can it not be alive? The answer lies in host dependency. A virus has the recipe for making more viruses, but it does not have the kitchen, the stove, or the ingredients to actually cook anything. To reproduce, a virus must perform molecular hijacking. It attaches to a living cell and injects its genetic material. It then hijacks the host ribosomes and enzymes, forcing that cell to stop its own work and start building new viral particles. Because they cannot reproduce or maintain homeostasis on their own, the biological verdict is very clear. Viruses are classified as obligate intracellular parasites, obligate intracellular parasites rather than living cells. To help you keep it straight for the exam, let's look at this quick comparison between living cells and viruses, which is shown in this table. When people ask why we study them in biology if they are not alive, remember they evolve and they have genes. These two important things. They evolve and they have genes. In the story of life, viruses are a special case that proves the rules are more complex than they seem. We have spent this lesson putting life into neat boxes, means kingdoms, phyla, genus, and species. But here is a secret most textbooks do not tell you. That is, nature does not care about our boxes. There are still limitations in our classification system. Human-made systems are just our best attempt to describe a world that is actually a continuous spectrum. As we have seen, there are always exceptions to the rule. For example, viruses are genetic fragments that act like living things but are not alive. Joglina acts like animals but feed like plants. These organisms prove that our boundaries are often arbitrary. Because evolution is a slow, gradual process, many species are caught in transition states. They are moving from one form to another over millions of years, making them nearly impossible to categorize perfectly. This is why we say science is a dynamic process. This is not static, this is a dynamic process. And classification is not a static list of names etched in stone. It is a working hypothesis. As our tools like DNA sequencing get better, our map of life must be revised to reflect the truth. So for centuries, we were like detectives trying to solve a mystery by only looking at the outside of a building. But with the genomic revolution, we finally got the keys to the front door. We moved from looking at physical shapes to reading the actual source code of life. The most powerful tool in this revolution is genetic sequencing. This is the process of determining the precise order of the four bases, which we earlier discussed, that is A, T, C, and G, adenine, thymine, cytosine, guanine, within a DNA molecule. But how do we compare a human to a bacterium? Then we look at ribosomal RNA analysis. So ribosomal RNA changes very slowly over a million of years, acting like a molecular timekeeper that allows us to trace relationships across the entire history of life. This molecular data has corrected many mistakes made by early taxonomists who relied only on anatomy. It eventually led us to move beyond the five kingdom system to the even broader and more accurate three domain system. So in the past, we grouped things by how they looked. Today, we group them by who they truly are, written in the language of their DNA. So, if the five kingdoms were not the final answer, then what is there? Let's look at the supergroups of life, that is, three domains. In 1978, a scientist named Carl Woese looked past the microscope and into the molecular clock of life. Ribosomal RNA, but what he found was shocking, very shocking. He discovered that some organisms we thought were just bacteria were actually as different from bacteria as we are. This led to the three domain system, which is the highest level of classification. So instead of five kingdoms, we now start with three massive domains that sort all life based on their fundamental molecular structure. So these are the true bacteria. The first one is domain bacteria. Okay, these are the true bacteria we encounter every day in the soil, in water, and even in our own gut. And the second domain is domain archaea. Okay, these look like bacteria, but their molecular chemistry is entirely different. They're often extremophiles, living in boiling volcanic vents or super salty lakes where nothing else can survive. And the last one is domain eukarya. This domain includes everything with a complex cell and a nucleus, including all the plants, animals, fungi, and protozoa, which we have earlier discussed. So, for your exams, you need to know the invisible differences between these three. So, by looking at this table and to memorize this table, you can easily differentiate and categorize these three domain system. Okay, if you remember, memorize this table, it will be very easy for you to ace your exams. Different features have been listed here like cell type, nucleus, membrane bound organelles, cell wall, ribosome size, DNA structure, histones means DNA proteins and their habitat. Okay, it will be very helpful for your exams. So this three domain system proves that life history is even deeper than we imagined. Now that we have mapped the entire tree of life, we shall move to our next lecture in my next video. Okay, that will be uploaded very soon, and I'm very hopeful that till this slide you have learned about classification systems, you have learned about the characteristics of life, you have learned about the differences between different living organisms. So stay subscribed and continue listening and watching these video lectures. See you very soon. Goodbye.